This is Amanda Welch welcoming you to this Bite Size Bio web seminar, which today is sponsored by Twist Bioscience. Twist oligo pools are highly accurate with an industry-leading 1 to 1,000 nucleotide error rate, allowing you to clone into expression vectors to generate libraries of single-guide RNAs. Our silicon-based technology allows for more representation, giving you efficient screens to accelerate your research. Enjoy accuracy now at the right price. Now, today's presentation is titled Discovering PARP Inhibitor Resistance Mechanisms Using Genome-Wide and Focused CRISPR Screens. It's being presented by Dr. Stephen Pettitt, who is a staff scientist at the Institute of Cancer Research in London. Stephen is now a staff scientist at the Breast Cancer Now Toby Robbins Research Center at the Institute of Cancer Research. He completed his PhD in 2010 under the supervision of Professor Alan Bradley at the Wellcome Trust Sanger Institute. During his PhD, he developed methods for high-throughput genetic screens in mouse embryonic stem cells. Stephen joined the group of Professor Alan Ashworth and Professor Chris Lord at the Institute of Cancer Research as a postdoc, where he used genetic screens to reveal the mechanism of action of PARP inhibitors and other inhibitors of components of the DNA damage response. He employs a range of cutting-edge experimental genetic approaches and DNA sequencing to investigate drug resistance and sensitivity in cancer, and uses genetic screens to identify synthetic lethal interactions with tumor suppressor genes. Now, as always, we will have a question and answer session after the presentation. So please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Stephen at the end. The recording of the webinar will be available at bit.ly/PARPwebinar. That's bit.ly/PARPwebinar, all one word, lowercase. So now over to you, Stephen, for the presentation.
Okay, thank you, Amanda. Welcome everyone. Thank you for coming to this webinar. So I'm Steve Pettitt. I'm working in Chris Lord's lab now here at the Institute of Cancer Research in London. So I'd like to start by thanking Twist and Bite Size Bio for this opportunity to present some of our work to you today. So I'm going to be talking to you about two different CRISPR screening methods that we've been using in the lab to study resistance to a class of drugs that we're particularly interested in here, which are called PARP inhibitors. So I appreciate there's probably a fairly wide range of backgrounds of audience here, so I'll introduce PARP inhibitors and CRISPR screens before we start the webinar. I'll start by explaining some of the background about PARP inhibitors and why we're interested in PARP inhibitors. So PARP stands for poly-ADP ribose polymerase, and there are various different PARP family members in the cell, but for the purposes of this talk and for generally when we're talking about PARP inhibitors in cancer, we're mostly interested in PARP1, which is the most abundant PARP in the cell. So PARP1 is expressed at very high levels in the nucleus, and it's involved in various different processes in the cell, but the one of most interest to cancer is its role in DNA repair. So on this slide, you can see this kind of cycle of PARP and how it acts as a sensor and a mediator of DNA repair. So if we start at the top here with point 1, you can see the protein structure of PARP1 is this kind of beads on a string model. So at the N-terminus here, we have this zinc finger DNA binding domain comprised of 3 zinc finger domains. Then in the middle, we have the BRCT domain and the WGR domain, which I'll come on to later in the talk. These are some domains important for the function of PARP1. And then finally, at the C-terminus, we have the actual catalytic domain of PARP1, which conducts this polymerization reaction. And there's an autoinhibitory helical domain, HD, here, which under more normal circumstances is folded up against the catalytic domain, inactivating PARP activity. So PARP's canonical role in DNA repair is as a sensor of single-stranded DNA damage. So PARP1 binds extremely fast to sites of single-stranded DNA breaks in the cell within sort of milliseconds. And the sort of first stage of binding is this the zinc finger domains, which assemble on the site of the single-stranded break. You can then see, as following the cycle round here, that the rest of the domains of PARP1 then fold up against the zinc finger domains. And this induces a conformational change in the helical domain, which activates PARP1. So now PARP1 can bind its substrate, beta-NAD, and start to produce these PAR polymers. So poly-ADP ribose, we usually abbreviate to PAR, and we refer to this process of polymerization as PARylation. So PARP1 produces PARP polymers on its substrate proteins, and these include DNA repair factors that actually carry out the repair of this break, such as XRCC1. But the other main activity of PARP1 is actually autoparylation. So PARP modifies itself, and this modification occurs on the BRCA domain and elsewhere. And it's thought that by modifying itself in this way and building up these long chains of poly-ADP ribose polymers, this results in a big steric buildup of negative charge, which promotes the dissociation of PARP1 from the DNA, and that allows this cycle to start again. And there are also PAR glycohydrase enzymes in the cell, which will remove this parylation. So this is on the next slide, just summarize this slightly more simply, that PARP is a sort of sensor and a mediator of DNA damage repair, particularly at sites of single-stranded DNA damage. So why is this important for cancer? So I work in a breast cancer research lab here, and we're particularly interested in the BRCA1 and BRCA2 tumor suppressor genes, which cause a hereditary predisposition syndrome to breast and ovarian cancer. So on this slide, I'm just going to explain the rationale for using PARP inhibitors in cancers with mutations in BRCA1 and BRCA2. So the original hypothesis was that if you inhibited PARP1, you would overload the cells with unrepaired single-stranded DNA damage. And then when the cells divide and go into S phase and a replication fork comes along and hits this single-stranded DNA break, this damage will then be converted into a double-strand break. And this is a particularly toxic kind of double-strand break because this is single-ended when it occurs at replication fork. There's nowhere obvious to join this break to. So these kind of replication forks that stall in this way absolutely require homologous recombination pathway to restart the replication fork and to continue replicating the cell's DNA and dividing. So this process is dependent on the BRCA1 and BRCA2 genes. So the thinking is that if you inhibit PARP1 and you accumulate a lot of unrepaired single-stranded DNA damage, normal cells will be able to repair this fine in S phase because they have an intact homologous recombination pathway. But if we have a tumor cell which has a mutation in BRCA1 or BRCA2, this cell will not be able to deal with this damage and will die. So yeah, the hypothesis was that inhibiting PARP1 would make the cells very, very dependent on a functional homologous recombination pathway. And so you can see some evidence of that in this slide from one of our lab's early papers on PARP inhibitors. So this is showing a survival curve of BRCA2 mutant mouse embryonic stem cells treated with a PARP inhibitor. And so what you can see is the BRCA2 mutant cells are very, very sensitive to this PARP inhibitor, about 100 to 1,000 times more sensitive than either the wild type or the heterozygous counterparts. So what that means is that this could be a potential treatment for BRCA2 mutant tumors because you'd be able to specifically kill the tumor cells which will have lost BRCA2 function completely, but not the wild type or the heterozygous carrier cells of the rest of the patient. Since these observations, PARP inhibitors have now been developed in both breast and ovarian cancer as treatments for cancer. But going back to this PARP catalytic cycle for a moment, there's another kind of implication of this cycle of PARP activity that I was explaining. So I mentioned that the original binding of PARP1 to the site of single-stranded DNA damage occurs via the zinc finger domains and doesn't require the catalytic activity. And also that the catalytic activity is required to dissociate PARP1 from the DNA. So that means that there's a kind of potential that you would end up with this in the presence of a PARP inhibitor, where you'll actually get PARP1 able to bind to the site of damage, but not to complete this cycle and dissociate. And this is kind of referred to as the trapping hypothesis of how PARP inhibitors work. So, perhaps when we treat with the cells with a PARP inhibitor such as olaparib, as well as not repairing this single-stranded DNA damage, we might end up with some kind of hypothetical structure like this, where we have PARP1 with the inhibitor bound stably associated with the site of damage. And it might be that this PARP DNA complex is very, very toxic to the cells. And some evidence for that comes from some previous work that we did, and also from lots of work by Yves Pommier's group, where we showed that if you do a transposon screen to try and isolate PARP inhibitor resistant mutants in wild type homologous recombination proficient cells, you often get resistant mutants that have lost PARP1 expression. And you can see that these mutants are very, very resistant to the cytotoxic effects of PARP inhibitors such as olaparib. So what this is telling you is that actually, if you delete PARP1 completely, the cells grow fine, and this is much less toxic than treating the cells with the PARP inhibitor. So this implies that PARP1 itself is required for much of the cytotoxic effects of PARP inhibitors, and that supports this hypothesis that PARP1 protein is actually a component of the toxic lesion, and it's not just the lack of PARP activity mediating single-stranded DNA repair that is killing the cells. So the final thing to introduce about PARP inhibitors is just some of the drugs that I'll be talking about during the rest of this webinar. So on the right of this slide, you can see 5 different PARP inhibitors, which are the most advanced clinical PARP inhibitory compounds. So 3 of these are now FDA approved for treatment of cancer. Olaparib, rucaparib, and niraparib are all approved for maintenance therapy in BRCA mutant high-grade serous ovarian cancer. And olaparib has also recently been approved for BRCA mutant triple-negative breast cancer. And in this talk, I'm going to talk mostly about olaparib and also about talazoparib, which is a new generation of PARP inhibitors. And talazoparib is an interesting drug because although all these 5 PARP inhibitors are quite good catalytic inhibitors of PARP enzymatic activity, talazoparib shows, relatively speaking, a much stronger potency to trap PARP1 at sites of DNA damage. And I'll show some more data on that later in the talk. And the other outlier on this sort of trapping potency scale is veliparib, which, although it's a good enzymatic activity, a good inhibitor of PARP's enzymatic activity, it seems to cause relatively much less trapping than the other drugs. And it's not really clear yet if this is sort of relevant in the clinic or not, but this is an interesting sort of differences between these drugs. So as I mentioned, we are seeing more and more patients now who have received PARP inhibitors as cancer treatments, both as part of the trials to develop these drugs and now also in routine clinical practice. And it's clear that resistance to these inhibitors will eventually develop in many cases. So far, the only kind of well-established clinical mechanism of PARP inhibitor resistance is by reversion of the BRCA1 or BRCA2 mutation. So what that means is these mutations that we see in patients in BRCA1 or BRCA2 are often frameshift mutations that put the protein out of frame, and sometimes in the resistance setting, you'll see a compensating frameshift mutation that will put the gene back into frame and restore homologous recombination function to the cell. So this is one way that we know cells can become resistant to the inhibitors, but there's also many unexplained cases of resistance as well. So that's really was the main question of this study that I'll be talking about today, is how can cells become resistant to PARP inhibitors? So I'll talk about kind of 3 approaches we've taken to that. So the first was a CRISPR screen to try and discover new PARP inhibitor resistance mechanisms. This was done in homologous recombination proficient cells originally, and we've since repeated that in BRCA1 mutant cells as well. So this was a genome-wide CRISPR screen and could potentially identify new mediators of PARP inhibitor resistance. And then in the second part of the talk, I'll talk in more detail about PARP1 mediated resistance and different mutations in PARP1 that can cause resistance. So that's the introduction to PARP, and now I'm just going to briefly introduce CRISPR mutagenesis. So again, I'm sure many of you have heard of CRISPR mutagenesis, and you'll be aware that this is a sort of 2-component system for experimentally introducing mutations in cells. So this consists of a bacterial nuclease, Cas9, which you can program with a short guide RNA, which consists of the 20 nucleotide gene-specific region, shown here in green, and this kind of hairpin scaffold region further down. And so when you have Cas9 complexed with this short guide RNA here, this will program Cas9 to sort of find the complementary sequence to this guide RNA in the target DNA, and Cas9 then introduces a double-strand break using its 2 nuclease domains at this target site. This break is then repaired by the endogenous cellular DNA repair pathways, and in many cases, this will eventually result in small insertions or deletions at the target locus. So in this talk, we'll mostly be providing this Cas9 machinery in the following way. So the guide will be provided on a lentiviral construct using a U6 polymerase III promoter to transcribe the guide from DNA. And the Cas9 is going to be stably integrated in the cells in most of the applications I'll talk about. So we'll infect cells with this lentivirus. This will program the constitutively express Cas9. This will then go and cut its target site hopefully on both alleles, giving us a mutant cell. And when we're doing screens, we'll actually use pools of lentiviruses designed to cut every gene in the genome. So this gives us a big pool of mutant cells with mutations in different genes, and we call this a mutant library. So this is what we can use as a starting point for our genetic screens. So this was the first screen that I carried out for PARP inhibitor resistance. So we took one of these mutant libraries. This is a genome-wide library in mouse embryonic stem cells, and we have 5 guides per gene on average. So altogether, there are about 100,000 guides in this library. And just did a very, very simple screen where you expose the cells to a high concentration of PARP inhibitor, talazoparib, it's also known as BMN 673, for 6 days. And this is at a concentration that would normally kill all of the wild type cells. So we just see what survives. And in this case, we pick the colonies, and then you do a PCR on the lentiviral construct, which is stably integrated in this colony, and then we just sequence it to identify which guide RNA was present in these cells that survived. So this is what we found when we looked at the surviving cells on this screen. So normally when we're doing genetic screens, you tend to, because we've got 5 guides per gene in this library on average, we'd normally look for multiple guides coming out in the resistant population, which will kind of give you some confidence that this is not a random effect or an off-target effect of the guides. So as we kind of expected from some of our earlier work, we saw a lot of PARP1 guides in the resistant cells from this screen. And we found 2 of the 5 guides total in the library, and everything else that we found was only represented by a single guide. So this was kind of this was expected to some extent, but when I went on to look at these mutants in a bit more detail, we found something interesting. So as you can see, many of these mutants lack PARP1 expression. So this is what we expected, but there's one exception here where even though this mutant clone 8 has a PARP1 guide, it's still expressing PARP1. So nevertheless, this clone 8 was very resistant to all the PARP inhibitors that we tested when we retested it. And you can see its resistance is shown in purple on these survival curves here. You can see that it's resistant to the same extent as a null mutant, which is shown in the green and the red here. So yeah, this is a clearly resistant mutation, and it's got a PARP1 guide. So what's happening here? So we went on to sequence the PARP1 target site in these clones, and this is just a PCR across the target site. And you can see in many cases actually you get quite large deletions at PARP1 in these clones, but in clone 8 everything looked normal until we sequenced this. And so here you can see the guide RNA sequence. So this is the target site. This is the PAM, which also specifies the target site for CRISPR. And you can see that we have just a deletion of 3 nucleotides at the target site in this mutant clone. And you can see that happens on both alleles, and then we actually have a heterozygous substitution mutation as well. So it seems like this is an in-frame mutation, which explains why it's still expressing PARP1. So what's happening in this mutant PARP1 protein? So this is deleting methionine 43 essentially, and then one of the mutations is causing a substitution as well. And when we looked at some of the crystal structures of the PARP1 zinc finger DNA binding domains bound to DNA, you can see that actually these 2 residues that are affected in this mutant actually form a crucial interface between the 2 zinc finger domains. So it's possible that this mutation could disrupt the DNA binding activity of PARP1. So that was our hypothesis, is that this mutant might not be able to bind DNA, and therefore it might not become trapped on DNA, and therefore we wouldn't see the toxicity associated with PARP inhibitors in these cells. So on this slide, I'm showing the trapping assay that we do for PARP1. So this was developed by Yves Pomier's group. So as you can see in wild type cells, when you add a PARP inhibitor, you see PARP1 becoming associated with the chromatin fraction when you do subcellular fractionation of the lysates in these cells. And this is all done in the presence of alkylation damage, which induces single-strand breaks. Whereas in our mutant here, you can see that all the PARP1 is remaining in the nucleus soluble fraction, and we never see any trapped in the chromatin fraction. So it seems like in this case, we've isolated a point mutant in PARP1 that can't bind DNA, and that therefore doesn't show this trapping effect and isn't toxic to the cells in the presence of PARP inhibitor. So this was quite a nice, I think this is quite a nice example of a CRISPR screen, because by isolating a point mutant like this, you can really get directly to the mechanism of action of PARP inhibitor drugs. So the next big question for us is, could PARP1 cause PARP inhibitor resistance in BRCA mutant cells? So this was always a experiment that I didn't think would necessarily work very well, because we know that deleting PARP1 and deleting BRCA are synthetically lethal. So the cells should not tolerate the simultaneous loss of both of these proteins. But having seen some of these point mutations coming out of the screen, I wondered if some of these point mutations might be tolerated in the BRCA mutant cells, and so maybe this could be a mechanism of resistance, even though the BRCA mutant cells should be very sensitive to the loss of PARP1. So I took a BRCA mutant cell line, a SUM149 breast cancer cell line, and we introduced the similar guide that we found from the screen, although this is now a human, targeting the human region of the orthologous region in human PARP1. And as you can see, that we see a really strong PARP inhibitor resistance phenotype when we transfect the cells with this guide. So clearly, the cells are able to tolerate PARP1 loss in this case and become resistant to PARP inhibitor. And we were able to clone out several long-term resistant clones from this population. And while some of them did indeed have very similar mutations to the mutation that we isolated from our screen, some of them are actually complete nulls as well. So it turns out this doesn't even depend on having a point mutation here. These cells were just able to tolerate the complete loss of PARP1. In a xenograft model, this also held true. So you can see here that while the wild-type xenografts respond to talazoparib and it improves survival, the PARP1 mutant xenografts do not. So so this was a question of why can these BRCA1 mutant cells tolerate PARP1? And we knew from kind of earlier experiments using kind of double RNAi experiments that these two things should be synthetically lethal. So I kind of repeated some of these experiments, but including our PARP1 null mutants that we identified from this screen. And what you can see from this experiment is actually, even in the PARP1 null mutant, if we knock down BRCA1 and BRCA2, we still see this synthetic lethality that's quantified over here. So it seems like these cells, even though they come from a BRCA1 mutant patient, they still have some residual BRCA1 activity, and it seems like that is allowing them to tolerate this mutation in PARP1. And our hypothesis for this, which is that there's a known splice variant of BRCA1 which skips exon 11, and that's where this frame shift mutation is in the SUM149 cells. And it's known as well from some other functional work that this isoform of BRCA1 which skips exon 11 still gives some homologous recombination activity to the cells. So this is our hypothesis. This was our hypothesis for why the cells were tolerating the loss of PARP1 in this case. So to test that a bit further, we used some of the other patient-derived cell lines we have in the lab. So in COF362, which is an ovarian cancer cell line with an exon 11 mutation, again, we were very easily able to isolate PARP inhibitor-resistant clones with a PARP1 guide RNA. But in MDM-B436 cells, which have an exon 20 splice site mutation, so this is not an exon 11, we could only ever get a kind of transient resistance to talazoparib, and we were never able to isolate any long-term resistant clones from these cells. And in fact, when we sequence the target sites in these cells, we always see at least 50% of the reads, even in this transient population, have wild-type sequence. So it seems like the complete loss of PARP1 is not being tolerated in these cells. So this is important because I mentioned that we know that reversion mutations happen in patients that become resistant to PARP inhibitors, and that's we have a cell model of that as well, which is shown in red here. So this is the SUM149 cell line with the BRCA mutation reverted, and in blue you can see our PARP knockout SUM149 clone, and you can see that actually this is even more resistant to PARP inhibitors than the revertant. So the question is, like, if we see mutation, if we see resistance emerging in patients by these mechanisms, would that make any difference to how we would treat the resistant patients? So as you can see here, this is showing the response to cisplatin in these same cell lines, and the revertant has also become resistant to cisplatin. So platinum drugs also target homologous recombination deficiency to some extent. But actually, as you can see, the PARP mutant here, it still retains the kind of wild type sensitivity to cisplatin, or maybe even a little bit more sensitive than the wild type. Similarly, if you look at topoisomerase inhibitors in these lines, the revertant cell line has become cross-resistant to these topoisomerase inhibitors, whereas the PARP1 mutants have actually become sensitive again. So this kind of suggests that knowing the mechanism of resistance in patients could be important for determining any future treatment that occurs. So moving on to the next part, I was interested in this question of exactly which PARP1 mutations could cause resistance. So this diagram of the PARP1 structure here shows the approximate location of the guides that we used, that were in our CRISPR library that we used to do the initial screen. So we have 5 guides, and as you can see, they're mostly towards the N-terminus, and that's a function of most of these CRISPR design algorithms will tend to design your guides towards the N-terminus because these are thought to be more likely to productively knock out your gene. But of course, obviously in PARP1, this actually misses what many would regard as the business end of the molecule, which is the catalytic domain. So I was wondering if, as well as the zinc finger domain mutations that we already found, whether we'd find interesting mutations in the rest of PARP1 that could also affect the function. So yeah, this is the guide that gave us the 45 DLM mutation, which had the zinc finger, which was the zinc finger DNA binding domain deficient mutant. But yeah, I was worried if we might be missing interesting mutations in the rest of the gene. So I designed this dense PARP1 library, which basically is just every possible guide that you can design across PARP1. Many of these may not be, the best guides or the most active guides, but I was going really for the best possible coverage we could get of the PARP1 gene using CRISPR. So this is where the Twist oligo pools really came in handy, because in the lab we're making a lot of CRISPR libraries now, and so using these oligo pools, you can easily sort of synthesize combinatorial way mixtures of libraries very efficiently in one of these oligo pools. So per pool you can get 72,000 oligos. This is only sort of about 500, so we can do basically lots and lots of different libraries in the single pool. And what we do is we just synthesize these. There are about 100 bases altogether. We have some PCR adapters, the cloning sites to put them into the lentiviral vector, and the guide RNA sequence in the middle here. And so then what we do is we just, for each library that we design in this pool, we do a sort of library-specific PCR with these PCR adapter sequences to convert this oligo to double-stranded DNA and amplify it just a little bit out so we can purify it from the rest of the pool, and then clone this into the lentiviral guide library. And this has proved to be a really efficient way of making lots of highly customized libraries now, such as this one to just target PARP1. And here you can see I don't think in this case we sequenced the actual plasmid that we cloned it into, but this is sequencing cells that have been infected with the PARP1 dense library that we made. And so you can see in most cases we get a good representation of all the guides in the library. In this one we missed a few at the cell stage, but you can see in this one all the expected guides from our design were present in the library. And you can see from this the Gini index here, this means that a low Gini index means that the counts are very comparable in this case. So we have a good representation, a good even representation of the guides in these libraries. So the other part of this experiment to look at which kinds of mutations we could see giving resistance in PARP1 was this cell line that we made. So this is a SUM149 cell line which carries a PARP1 with a knock-in of GFP into the endogenous PARP1 locus. And we did this using the CRISPR system. I didn't put the reference here, but you can look up the CRISPR system, which is a very easy way to knock in tags into the C-terminus of a gene of interest. So yeah, in these cells we have PARP1 GFP being expressed from one of the endogenous PARP genes, and SUM149 cells are a little bit aneuploid, so I think there are actually about 5 copies of PARP1 in these cells. So then the idea is we could introduce our PARP1 guide library here and then select the cells for talazoparib resistance. So that means that we should get a loss of function of PARP1. And there's 2 possibilities for how that could occur. Either we'll get a complete knockout of all the PARP1 alleles in the cell, and they'll become resistant to BMN. So BMN is talazoparib. Or we could get mutations like we isolated from the screen that preserve the reading frame of PARP1. So these would be small, hopefully in-frame deletions. and these cells would still express the GFP. So so what we did was we put in our guide library, we selected Thalassopterid, and then we sort out GFP positive cells. And then, so this gives us resistant cells that still express in-frame PARP1 GFP. And then what we do is we reverse transcribe from the GFP and sequence the PARP1 gene using amplicon sequencing. So this is a diagram of the PARP1 gene again. You can see the domain structure here. And this is, again, the positions of our guides along this site. So the outcome of this sequencing experiment is basically we call the mutations and we map them onto the PARP1 sequence. And so what you can see is we see some interesting clusters of mutations occurring in this sequencing. So we're just basically plotting the frequency of mutations at each position all along this all along the PARP1 gene, whether these were deletions or deletions or insertions. So as we kind of expected, we see clusters in the zinc finger domains. So zinc finger domain 1, which is where we found the original mutation in the screen. Also zinc finger domain 2, lots of mutations. Then we don't see very many mutations in the BRCT domain, and that's sort of, I guess, consistent with what we know about the function of the BRCT domain. This is where the auto-modification occurs, but it's probably not that important for the DNA repair functions of PARP1. And then we see a lot of interesting clusters in the WGR domain here, and also in the regulatory domain. And again, not very much in the catalytic domain, except for this one sort of hotspot here, which is not the active site. And again, this is probably expected to some extent, because you'd expect mutations that disrupt the catalytic domain to result in constitutively trapped PARP1, which might be very toxic to the cells. So the next thing we did was actually to map these frequencies of mutations onto the 3D structure of PARP1. And here you can see really nice patterns. So if we look at this crystal structure here with the zinc finger domain, with the zinc finger domains 1 and 2 bound to a single strand break, you can see that on these 3D structures, we've colored the residues red and sized them based on the frequency of mutations that were identified in the screen. So you can see actually that the frequency of mutations really nicely identifies the DNA binding interfaces of the zinc finger domains. So we're not just seeing mutations occurring everywhere in the resistant population. They seem to be, it seems to be these ones that actually contact the DNA that are really important. And that, if we look at these other crystal structures, which include some of the other domains of PARP1 as well, you can see that kind of also holds true for some mutations on this side of the WGR domain, which is also contacting DNA. And then we also see this big bundle of mutations in the middle of the WGR domain going through to the helical domain. And our hypothesis is that this is a very important region for transmitting the activation signal from the DNA binding of PARP1, so its sensor role in sensing DNA damage, through to the catalytic domain, which is required for the DNA repair. So this, I think this very nicely identifies the key functional areas of PARP1 for PARP inhibitor action. And so the other side of this project is obviously we're very interested to see if PARP1 mutations occur in resistant samples. And some of our collaborators in Seattle, Liz Fisher and Jungming Lee at the NCI, had identified one mutation in a PARP inhibitor resistant patient from a phase 1 trial. This is an R591C mutation. So this is a missense mutation, and it was kind of a variant of unknown significance. But when we mapped it onto our mutation data from this screen, you can see that this R591C mutation really fell into one of these WGR domain clusters that were highly mutated in the resistant population, suggesting that it might indeed be very important for the function of PARP1 and for trapping. And if you look at that on the 3D structure, again, you can see that R591C is really right in the middle of this this bundle that we think is involved in transmitting the signal to the catalytic domain. So yeah, that suggested that this R591C mutation may actually be deficient in trapping or in DNA binding. So Drago Krastev, who's another postdoc in the lab, tested this using a microirradiation assay. So in this assay, we shine a UV laser onto PARP1 GFP expressing cells to make a little spot of DNA damage in the nucleus. And then using live microscopy, you can monitor recruitment of GFP signal to this spot that you irradiate. So if we look at this graph here, in black, you can see the normal wild type kinetics of PARP1 binding. So PARP1 comes on very, very fast within a second, and then dissociates a little more slowly as DNA repair occurs. If we add talazoparib, shown in the gray, PARP comes on and stays on. So this is the trapping effect, which you can see very clearly in this microradiation assay. But if you look at the colored lines here, which are showing cells expressing a GFP fusion with the R591C mutant, you can see that although these do bind, they're not stably bound at all, and both in the presence or absence of talazoparib, these mutations quickly dissociate from the site of DNA damage. So this suggests that really these are not able to bind stably to sites of DNA damage and to activate PARP1 and mediate repair. And also, you can see from this graph that adding the talazoparib doesn't cause trapping in these mutants. So that really suggests that actually it is a trapping DNA binding defect in this R591C mutation that might have been responsible for the lack of response to PARP inhibitor in this patient. So that's what I was going to say. So just to recap the talk here. So I've shown you that PARP1 DNA binding is really required for PARP inhibitor cytotoxicity. That's in both HR proficient cells and also deficient cells. So this is shown by the isolation of PARP1 loss of function and DNA binding domain mutations. And yes, surprisingly, even in BRCA1 deficient cells, this was a possible mechanism of resistance. And that was because some of these pathogenic BRCA1 mutations will actually tolerate PARP1 loss or PARP1 mutation because they are, in fact, they seem to be hypermorphs. And that's something that you can only see really when you use the patient-derived mutations for BRCA1. If you try and do knockouts in BRCA1, you won't see PARP1 coming out as a mechanism of resistance. And again, this was shown recently in another genome-wide screen we've done for PARP inhibitor resistance, where if you use a complete knockout BRCA1 model, you won't see PARP1 coming out as a mediator of resistance, presumably because it's lethal in this context. And so from the models that we isolated from these BRCA1 mutant cell lines, we showed that actually the resistance is very distinct from the kind of resistance you get from that occurs by reversion of the homologous recombination defect. So, for example, we don't see this cross-resistance to cystatin or to other DNA repair targeting drugs like topoisomerase inhibitors. And finally, from this last part of the talk, I showed that although the zinc finger domains are very important in mediating DNA binding and trapping, there's actually several significant clusters of mutations outside the DNA binding domain that can also affect trapping and affect resistance. And we think those are involved in transmitting the activation signal to the catalytic domain. But there are also other interesting clusters here, which might be which we're now interested in looking at further. Oh, and most of this work I've talked about today is published in the references down here, so you can read about it if there's anything else that you're interested in. So just to end by saying thank you to my hosts again here, Twist Bioscience and Bite Size Bio, to everybody in the lab who helped with these experiments, to my PI, Chris Lord, Drago Krastev, who's another postdoc who helped me with a lot of this work and did a lot of the microradiation assays in particular. Feifei, Jess, and Inga, who did a lot of the cell culture and model making involved in this project. I have to say a big thank you to our collaborators as well, Liz Swisher and Jungmin Lee, who analyzed their PARP inhibitor trial data for us to see if they could find any PARP1 mutations. And finally, also to Kosuke Yusa from the Sanger Institute, now in Kyoto, who provided us with the genome-wide CRISPR library and helped us out a lot. So thank you for listening, and I'm happy to take any questions now from Amanda. Thanks.
Thanks, Stephen. That was an excellent presentation. We have a few questions from the audience. If anyone else has a question, please feel free to post it in the questions box that appears on the right of your screen. So our first question comes from Monica, and they're asking about the reference for the GFP knock-in. Is that the reference that you were talking about on, I think it was just your last slide?
The reference, it's, I don't remember it off the top of my head, but it's a Nature Communications paper, and the system is called CRISP Paint. So if you look up CRISP Paint, you should be able to find it. So this works by introducing, by sort of you transfect a plasmid with the tag in, and also guides that cut the plasmid and cut your target locus of interest. And the integration occurs, I think, by non-homologous end joining into your target locus. And we see some locus-specific effects on this, but in many cases it seems to be very efficient. I think we had, and obviously it's very easy to see if it's worked or not because your cells go green.
I love the title, Crisp Paint. So our next question is, are there PARP1 mutations that are seen in human cancer?
Yes, so PARP1 is not really an oncogene or a tumor suppressor gene in itself. So you don't really see a lot of mutations in primary tumors, and I don't think PARP1 is really involved in the development of tumors as such. So what we're looking for here really is acquired resistance in the most part. So surprisingly, this R591C mutation that we looked at was actually a pre-existing somatic mutation in the cancer, and this was found before the exposure to PARP inhibitors. This was in a phase 1 trial, and this patient was de novo resistant to PARP inhibitors. So yeah, we're really interested in looking more at acquired resistant samples to see if there are any mutations that occur during treatment. Because they don't seem to really be there in primary tumors.
Okay, and then to follow that up, Monica asks if you have any idea if there's any PARP1 inhibitors that are used in, say, pancreatic or other cancers.
Yes, so there are PARP inhibitor trials involved in going on in several other cancer types now. Pancreatic cancer is one for sure. Pancreatic and also prostate cancer. Any sort of in many cancers where you see BRCA1 or BRCA2 defects, or in certain other DNA repair genes, these are being considered as target populations for PARP inhibitors.
Okay, and so our next question is, are reversions the most likely cause of PARP inhibitor resistance?
So far, reversions have been the main mechanism observed in the clinic, and I think there's several reasons for that. First is, I think in a lot of cases people only look for reversions because common kind of tumor sequencing panels will include things like BRCA1 and BRCA2, but they won't include things like PARP1 because this is not an oncogene or a tumor suppressor gene, so it doesn't generally appear on these panels. The other thing is, I've showed in this talk that the reversions also cause cross-resistance to cisplatin in particular, and in many cases, patients on PARP inhibitor trials have been heavily pre-treated with cisplatin, particularly in ovarian cancer and also increasingly in breast cancer as well. So it's possible that resistance is actually developed in response to the platinum treatment, but then also applies to the PARP treatment. So it's only really now we're getting PARP inhibitors beginning to be used in early stage trials. So I'm interested to see if there's going to be any differences in the resistance mechanisms we see if PARP inhibitors end up being used before platinum, for example.
Yes, and so I think that kind of leads into, or that might have answered my next question, or the next question that we have about are PARP1 mutations a potential cause of resistance in other PARP inhibitor sensitive backgrounds?
Potentially. So it's been, I think one setting it has been shown in is in Ewing sarcoma cell lines. So Ewing sarcoma cells are sensitive to PARP inhibitors due to these EWSR1 fusions that occur in Ewing sarcoma, and these probably do induce some kind of homologous recombination defect, but it's somewhat distinct from the BRCA defects. But knocking out PARP1 in these cell lines does cause resistance. So I think provided the cells will tolerate the loss of PARP1, this will be a cause of resistance because it's acting very sort of close to the drug, if you see what I mean. Deleting PARP1 is really preventing the toxic lesion from ever occurring. It's not about how cells deal with it. So it's a very kind of upstream cause.
And then I believe our last question about is it necessary to use the GFP fusion when conducting focused screens?
Yeah, so we used it to kind of as a kind of trick to make our life easier in this case, because you can kind of sort out the in-frame fusions and enrich your resistant population for those. But I don't think it is necessarily going to be the case. I mean, you could just filter out your in-frame mutations of interest bioinformatically. Recently, we've also been using some of the CRISPR editing enzymes, which cause missense mutations rather than deletions. And again, in that case, you might be able to get away without. But one thing you do have to do in this situation is knock out all the alleles of PARP1, because we know even one PARP1 allele in the cell will be sufficient for the cell to be vulnerable to the PARP inhibitor. So
you just need one. Okay.
Yeah. So in this case, the GFP also helps, because we make sure we only look at the GFP allele, and we check. Yeah, and we assume that all the rest have been knocked out. And actually, in some of the clones we isolated, we have checked that, and we do see that all the other alleles in the cell have accumulated loss of function mutations.
That makes sense. Well, fantastic. So that looks like that brings us to the end of the webinar. So thank you again, Stephen, for a very illuminating presentation and a great question and answer session.
Thank you.
And thanks also to our sponsor, Twist Bioscience. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you've enjoyed the webinar and would like to view the video recording of the session, please visit the webinars page on bitesizebio.com. It should be available within the next 24 hours. And there, you can also see the other webinars we have lined up for you on Bitesize Bio. So until next time, good luck in your research, and goodbye from all of us at Twist Bioscience and Bitesize Bio.